Buenas tardes, mi nombre es Ramón Latorre, soy un neurobiólogo y Premio Nacional de Ciencias Naturales aquí en Chile, y quiero darles la más bienvenida a esta nueva actividad de Puerto de Ideas Valparaíso. Y Puerto de Ideas Valparaíso de verdad porque estoy en Valparaíso. Y vamos hoy día a una actividad que llamamos de moscas y relojes biológicos, en la que conversaré con Michael Rosbash. I hope I pronounce your last name correctly, Mike. I'm sorry.
Yes, no.
And he's a cronobiólogo estadounidense y premio Nobel de Fisiología o Medicina, lo cual él dice que es fisiología, sobre sus grandes descubrimientos en torno a los ritmos circadianos. Este año, Puerto de Ideas Valparaíso celebra su 10º aniversario gracias al apoyo de Colbún, Copeuch, Fundación Mar Adentro y el Ministerio de las Culturas. Esperamos que lo disfruten. Muchas gracias por acompañarnos y no se pierdan las actividades de Puerto de Ideas Valparaíso nos trae este fin de semana. Mike?
Yes.
Thanks for being here. Thanks for joining us.
The pleasure is mine. I guess I should begin, right? So it's a great pleasure. Todo el placer para mí. And I'm going to try to tell you a story today of this story of biological clocks and how it came about. And so the question is always, this is Alicia en el País de las Maravillas, si me recuerdo bien la traducción en castellano. So where should I begin? And the answer is always begin at the beginning. And of course, I haven't come to the end yet, but I will begin at the beginning and tell you where all this began. And so all this began a long, long time ago, when life really arose on Earth. And this inexorable, continual rotation of the Earth, the light-dark cycle that life has always been exposed to, is really what began as the original environmental cue that was critical for the origin of circadian rhythms. And we know today that probably the very first circadian clock arose in cyanobacteria. And this photosynthetic organism was responsible for the oxygenation of the atmosphere more than 2 billion years ago. And the world before this oxygenation event was a very different place with a reducing atmosphere. And one of the key ingredients required for contemporary life to arise was the much greatly increased concentration of oxygen. And so let me define circadian rhythms for you so everybody understands this. The word comes from the Latin, or circa dia, which means about a day, más o menos. And the reason we say about a day is because the endogenous periodicity of animals is not precisely 24 hours. It's a little bit greater or a little bit less than 24 hours. And the purpose of this timing event is to anticipate what's going to happen in the outside world. So we say in English that the early bird gets the worm. And it's also true that the early worm can avoid being eaten by the bird. And so that is really, anticipation is really the name of the game, if I could call it that. And the way we get to be exactly 24 hours, despite not having a precise 24-hour timekeeper, is by entraining to this light-dark cycle. And that entrainment event, the resetting of our not very precise 24-hour clock, occurs every day and is what keeps today the same as yesterday and tomorrow the same as today. And so an illustration of this phenomenon is shown here, where you see a rodent, a hamster probably, that's running on a running wheel, and the activity events of that animal are being recorded. And you can see how old the slide is, because instead of using a computer, there's a chart recorder, which is perhaps 40 years old. And when that rodent runs on the running wheel, the activity events, when in constant darkness, are indicated here, and rest or sleep is indicated here, where there are no marks. And you can see that every day, the activity begins a little earlier than the day before, and the activity ends a little earlier than the day before every day. And that's because this animal has an endogenous period of less than 24 hours. But if that animal is now placed in a light-dark cycle with 12 hours of daylight and 12 hours of nighttime, then every day is exactly the same as the day before, and the animal now has an entrained period of exactly 24 hours. This is what happens to us in the real world. You'll notice also that this animal is nocturnal. That is, it's active at night, and it sleeps or rests during the daytime. Okay, so how do we actually do this with fruit flies, with my favorite animal, Drosophila melanogaster? Well, the animal is, whoops, I'm having a little problem here. The animal is placed in a glass tube with some food on the bottom, and that glass tube is placed in a recording device. And this is an old version that actually is the version we gave as a gift to the Nobel Museum, in which the tube is flanked by a light source and a photoreceptor on the other side. And every time the animal runs back and forth along the tube, there's an event recorded. And that's the equivalent of the chart recorder I showed you for the rodent. And what's relevant here is that the light is in the infrared, and fruit flies cannot see in the infrared. So the light does not disturb their activity. The animal thinks it's in constant darkness. And now, almost 50 years ago, Konopka and Benzer, working at Caltech, identified these wonderful mutants of fruit flies, which had either short periods, long periods, or were completely arrhythmic. That is, that their locomotor activity patterns were very strange compared to the normal, almost 24-hour rhythmicity of wild type or normal animals. And you might ask, what's the purpose of genetics in this particular context? And it's not, as we usually think of genetics, of whether you are different from me, or perhaps to address nature-nurture questions. That is some trait we have is the fact that I am tall and Ramón is shorter than I am. Is that genetic or is that environmental? It is really as a possible entree into this mysterious process of circadian rhythms. What is the nature of the timekeeper? What is the quartz crystal, or what are the gears that are ticking away in this endogenous circadian timekeeper? And genetics allows you to enter into the problem and perhaps find some of the components and thereby try to address the nature of the timekeeping. And of course, this relies on not only genetics, but then ultimately on sequencing the genes and the proteins that are involved in the process. And so I was encouraged to tell you a little bit about my personal history. And I should say that I arrived at Brandeis as an assistant professor at the age of 30. And I didn't know anything about neuroscience. I never had heard of circadian rhythms. I didn't know what they were. I was a biochemist who worked on nucleic acids. And I met, this is me, and I met Jeff Hall, who was a assistant professor in the biology department, just like I was. And we became very good friends. And it's really through Jeff that I learned about genetics, I learned about fruit flies, and I learned about circadian rhythms. And we formed a partnership and worked together for more than 20 years. And you can see here us as young men engaging in two of our shared vices, namely tobacco, a cigar in my case, and alcohol in Jeff's case. And so throughout the 80s and 90s, we engaged in lots of work, genetics, cloning of genes, recombinant DNA, biochemistry work, and much of this work was done independently by Mike Young's lab at the Rockefeller University. And this work led to a model of how the circadian timekeeper might work. And that model, which is depicted here and was finalized more than 20 years ago and is still true today, indicates that there is a transcriptional feedback loop, that there are proteins which drive the synthesis of other proteins, which are actually negative regulators of their own synthesis. And eventually they feed back and turn off their own synthesis. So this is negative feedback loop are key features of oscillators in electronics as well as in biology. And this very simple feedback loop of macromolecular metabolism of transcription is really at the core of circadian timekeeping. And as you would infer from what I've said, it takes 24 hours, about 24 hours, circa 24 hours, to go around this cycle to both synthesize these proteins and then for those proteins to inhibit their own synthesis, to turn off their synthesis, and then for the proteins to decay and for the whole process to begin again. And it turns out, through our great fortune, that what we had discovered in fruit flies, as Ramón told you, if I understood his Spanish correctly, that it was subsequently found that the same mechanism, the exact same process, and even the same proteins, the same genes and proteins function in other animals and even in humans. And that, of course, turned out to be important for the general acceptance of this process. And I should say that I'll tell you a little bit about fruit flies and model organisms in this context. And this cartoon was made by, this is Mike Young, this is Jeff Hall, my colleague at Brandeis, and this cartoon was made by a grandnephew of Mike Young's, and it is to illustrate the debt we owe to fruit flies. And so despite the fact that Nobel Prizes are limited to 3 human beings, we thought maybe fruit flies could be a 4th recipient and could share the prize with us. Now, the reason I say that, and something you probably don't know, is that our Nobel Prize was the 5th Nobel Prize that was given for work that was done with fruit flies, beginning with the great genetic pioneer, T.H. Morgan, who won the first genetics, first American physiology or medicine Nobel Prize in 1933. And since that time, of course, there have been 5 Nobel Prize winners based on fruit flies. And this organism is being used today to model and study many diseases, as indicated here: cancer, drug addiction, aging, many, many different processes. And of course, the reason that this is so popular is because they're much less expensive than mice. The genetics is outstanding. They have a short generation time, small genome size, all features which make them an outstanding experimental organism. You probably don't know that organs and hormones are conserved between fruit flies and humans. For instance, the digestive system. There are kidneys in fruit flies that are very similar to what happens in humans. Hormones, insulin, glucagon, neurotransmitters, such as molecules that Ramón is interested in. These make fruit flies really excellent model organisms for studying these processes. And even genes, which are responsible for human diseases, 80% of these genes have been identified in fruit flies, and they do exactly the same job in fruit flies as they do in humans. So all of this speaks loudly for the importance of fruit flies in basic research today. So let me tell you a little bit about the effect of circadian rhythms on human physiology. So why is circadian biology so important? And it turns out the reason is because this clock, which I illustrated before, this transcriptional feedback loop, drives the rhythmic gene expression of hundreds or thousands of genes wherever it's present. So it's not just this little loop which is functioning, but this loop sits on the top of a gene expression hierarchy so that a large number of genes in each cell or tissue, wherever that feedback loop is present, are undergoing circadian rhythmic oscillations. And of course, it turns out that that little feedback loop is present in almost every mammalian tissue. It's present in the brain, it's present in spleen, in the heart, in lung, and muscle, in kidney. It's present effectively everywhere. And as a consequence of these two facts, more than 70% of all genes in mammals, including humans, are expressed rhythmically in at least one tissue or cell type. And of course, that explains why circadian rhythms affect so much of physiology, because it affects the expression of so much of our genetic material. And so the question is, moving forward into the future, can we leverage what we've learned about circadian rhythms to affect or to improve human health? And I'm just going to touch on a couple of specific features. What's listed here are phenomena which are intimately impacted by circadian biology. But let me just say a word first about metabolic diseases, and of which diabetes is perhaps the best known example. And so this paper, which was published a half a dozen years ago or so, described a very interesting finding. And that finding was that if you feed an animal in a time-restricted way, so that, for example, that animal eats for 10 hours a day and then fasts or does not eat for 14 hours, that time-restricted feeding prevents metabolic diseases in animals that are fed, for example, a high-fat diet, which would normally produce metabolic syndrome or even diabetes. And so it turns out that despite the fact that the animal doesn't consume any more calories than it would normally if fed ad libitum, that is, eating whenever it wants, and despite the fact that its metabolism, its rate of metabolism doesn't change, something happens in vivo in the animal so that it does not get obese and it does not suffer metabolic syndrome. And so the lesson of this story is that it turns out what's important is not only what you eat and not only how much you eat, but also when you eat. And Ramón can perhaps ask me in the questions how we might interpret this in a more mechanistic or a more biochemical framework to explain this phenomenology, and I'll attempt to do that in a little more detail if encouraged. So the second thing I want to discuss for a few minutes is sleep disorders and sleep in general. So the circadian clock, as I mentioned to you, has an impact on all kinds of physiology, cardiovascular disease, asthma, learning and memory, but it also has a major impact on sleep, both when we sleep and how we sleep. And thank goodness, gracias a Dios, that slide worked correctly. So, and here's the current way in which we conceptualize the regulation of sleep. Sleep really has two main inputs into its regulation. One is circadian regulation, that is this metronome-like timing which we all experience every day, and that is why we get sleepy at 10 o'clock at night, and why we are alert in the morning, and why even we get a little bit sleepy in the middle of the afternoon as well, because the circadian biology is impacting our alertness. But there's a second regulatory system which we understand much, much less well, the so-called homeostatic regulation. And that system is keeping track of how much sleep we have had and how much sleep we need. And that is unknown, how that actually works. Who is keeping track of sleep need, which of course impacts how much sleep we experience. If you stay up late and are tired, then you sleep more deeply and longer the next night, except of course for old people who have trouble with all kinds of regulatory systems. But for healthy young people, that is their experience. And of course, we know that sleep is really important. If people are sleep deprived, there's actually a form of psychosis or PTSD, post-traumatic stress disorder, that occurs. And so this leads me to tell you a little bit about this interface between human rhythms and sleep. And by that, I will also tell you a little bit about people who are morning birds or larks on the one hand, people who wake up early, and people who are night owls on the other hand, who can't go to bed until quite late and then like to sleep quite late. And all of this is framed in the context of the fact that our endogenous rhythm, the endogenous rhythm of human beings, is about 24 hours and 15 minutes. So in other words, our clock runs about 15 minutes slow every day. And of course, as I told you, it's light, which resets that clock every day back to 24 hours specifically, perfectly. And morning birds run a little bit less than 24.25 hours, and night owls run longer than 24.25 hours. And I can illustrate something here important about a night owl and also introduce this concept of social jet lag. And these are some real data of a human being and that human being's sleep schedule for 5 work days in the pink barber pole-like stripes here. So this is Monday, Tuesday, Wednesday, Thursday, Friday, when that individual went to sleep and when they woke up. And then what happens on Saturday and Sunday on the weekend. And here's the next week, Saturday, Sunday. Here's the next week. Here's the next week, et cetera, et cetera. Now, what you notice here is, of course, this individual always wakes up at about 7 in the morning, here is shown here during this week, or almost always. And of course, that's because he or she uses an alarm clock to wake up, as many people do, in order to go to school or work. And of course, you'll also notice that on free days or weekend days, in general, that person goes to bed later and sleeps much later. And this distinction between the mid-sleep time during the work week or school week and the mid-sleep time on free days is called social jet lag. Because of course, the individual is experiencing continuously jet lag by virtue of changing its sleep structure 2 out of every 7 days, because it's either sleep deprived or has had its circadian rhythm misaligned. And it turns out that early birds, people who get up early and go to bed early, actually experience about 2 hours less social jet lag. So they're only about 1 hour different between free days and work days than night owls, which are about 3 hours different between free days and work days. Nope. Whoops. And of course, And we all know that social jet lag, or the difference between work days and free days, is maximal during the teenage years. This is when our children happily sleep till 2 or 3 in the afternoon when given the opportunity. And yes, happily sleep much more when given the opportunity. And this difference is probably due to sleep deprivation, but may also be caused by a misalignment of the circadian program with respect to the light-dark cycle. So probably those teenagers, and even most of us to some extent, are always sleep deprived by missing a little bit of sleep every night and being woken up by our alarm clock. And we know, of course, that young people are more owl-like than adults, as I indicated before. And this has sort of major implications for what time school should start and for daylight savings time. In other words, the students should be going to school later. And it's because of the teachers, at least in the United States, that school still begins relatively early. Teachers are adults and need to have a more programmed early schedule. And so I want to posit for you that in addition to these practical indications of organizing sleep, there's also a challenge, a basic science challenge for the future, and that is understanding the basic science of sleep. And as I indicated before, we don't know where or what is keeping track of sleep need, how much we need. In other words, how does sleep homeostasis actually work? And secondly, and related to this question, why do we actually sleep? We spend a third of our life doing this, and yet we actually don't know the deep conserved function of sleep. And I should point out to you that fruit flies really sleep, and they sleep very similarly to the way that humans sleep. For example, it's harder to arouse a fly when it's sleeping, just like with an animal or with a person. There's a quick reversal to wakefulness. The molecules that we know are involved in sleep, for example, stimulants or hypnotics, sleeping medication works very similarly in fruit flies as in humans. If you sleep deprive a fruit fly, it sleeps longer and more deeply the next night after being sleep deprived. So flies really do sleep like we do. And so there's a belief in the community that I share that maybe fruit flies will do for our understanding of sleep something similar to what they've done for circadian rhythms, namely lead the way into understanding, and maybe even before the problem is really cracked in mammals. So I've come to the end of my presentation in pretty good time, actually, since I have a timer going here. And I want to thank my current and former lab members. This is, of course, not all my former lab members, but they're recently departed lab members. And you probably need to understand, or many of you do, that doing life science research is a team endeavor. And I stand here as a prize winner and give you this talk, but in fact, it's really due to the efforts of many, many people over the decades, students and postdocs, that have contributed to the work that my laboratory's been able to do. And I owe really everything to their efforts. And so with that, I'll end, and I'll be happy to have a conversation with Ramón about work as we move forward.
Thank you, Mike, for your excellent talk. Very interesting. And I would like to say a few words why I am here.
I know.
And the story goes that in 1977, I arrived to Boston, to the Harvard Medical School, and I met a very good friend of mine at Brandeis, and his name was Chris Miller. So year passed, I returned to Chile, and Chile was undergoing a difficult situation. We had a dictatorship, and there were an immense amount of extremely good students, but the university was in shambles. And then some of them approached me. And what I want to say is that Brandeis and the Brandeis scientists, in particular Mike and Chris Miller, were extremely generous with Chilean students. And two of those Chilean students, Vivian Butnik and John Ewart, had the very fortunate opportunity to work and to be near, Mike and Jeff Hall, the other Nobel Prize. So that's the reason why I'm here. And I really thank you, Mike, for the generosity of Brandeis and you and Jeff.
Well, Vivian and John were fantastic students.
Okay, so
Mi esposa también, you know.
I know, that was my last question.
Okay, I'm sorry.
I can start with that. Okay, Mike. So your wife is Chilean, but is also a scientist, okay? So the first question is where and how you met? You met, and the second question is because you are both are scientists, if you take work at home.
So we met because she did her PhD in my laboratory. So she was my PhD student. This is this is many years ago. In fact, she came to Brandeis in 1979. So, same era, more or less, as you. And she is retired now. So she retired a long time ago now. And so I work from home, but she is also works at home, but she's no longer doing science.
Well, so let me start. Well, what you say is that Drosophila has played an extremely important role in genetics and neuroscience, and also it has been fundamental in the ion channel research. So, and then at your banquet speech, when you got the Nobel Prize, you tell us that this is the 5th Nobel Prize given to this fly. Okay, can you give a sort of a very brief historical account why this is so, and why this little fly is so important as a biological model? Why not to use rats or I mean butterflies, for example?
Well, so the landscape is changing very rapidly, and CRISPR is allowing for animals to be explored in a way that wasn't possible before. But for a very, very long time, genetics and the ability to manipulate genes and proteins and ion channels was almost unique in fruit flies. So people understood, or I should say some people understood, that the ability to manipulate physiology with genetic tools and even the ability to manipulate ion channels in vivo was unparalleled. And so certain kinds of experiments have just led the way all the way through. And the frontier scientifically was always being pushed in flies, I want to say, before the same kind of experiments could be done in animals. So transgenic animals, the ability to make an intact animal with an altered ion channel, for example, that was first done, and for years before such an experiment was possible in mice or rats. And so I think it was just the art of the possible. And of course, there's a tendency for, I think this all frankly comes from human exceptionalism. In other words, for thinking that humans are so exceptional and so mammals are exceptional. But the tremendous lesson from DNA sequencing from the early 80s, when organisms were started to be sequenced, was the fact that we're all the same. And of course, if E. coli, bacteria don't have a nervous system, so certain problems simply cannot be addressed in E. coli. But I think there was always a tendency for finding the simplest animal you could that would express the properties you're interested in, and then study that animal in the hope that you would be able to do something significant and quick. So I think that's the reason, and the biology kept rediscovering that fruit flies had really the same system. I mean, one of the Nobel Prizes for innate immunity, who, and of course, it's very important in this era of COVID, that is the nature of the immune system. Who would have imagined that fruit flies would have a very similar innate immune system to human beings, right? But they do. So, that's I think the lesson of reductionism. Take the simplest thing you can find and try to see what you can learn.
So if I remember correctly from my biology 101, Morgan was the first Nobel Prize.
Yes, he's the first American. Sorry, he's the first genetics Nobel Prize and the first American Nobel Prize in physiology or medicine. He's not the first physiology or medicine prize, but he was.
Yes, and what he discovered at the time.
Morgan?
Yeah.
Morgan and his team were able to map genes on a chromosome and shown that genes had a physical relationship with the chromosome. So in other words, the nature of inheritance after Mendel was really, nobody knew what a gene was. It was clearly it clearly it transmitted traits which Mendel could show in the middle of the 19th century. But until the beginning of the 20th century, and then there were some experiments that intimated that chromosomes also were involved in inheritance somehow, because it was clear by looking at cells that the chromosomes would divide, and if the cells would inherit a healthy complement of chromosomes, then they would be normal. And if their complement of chromosomes was abnormal, then of course they were abnormal. But but Morgan was able to put these two concepts together and actually also show that genes were distributed on a chromosome in a quasi-linear manner. So genes A, B, and C, B lay on the chromosome between A and C, and that gave them a physical characteristic. They had to actually be a physical entity that was connected to chromosomes. So that was really a pioneering insight into the nature of the gene and genetics.
Mike, I am very curious. When you discovered the period gene, did you think that finding would lead you towards the Nobel Prize, or was so important that, I mean, like hitting the ceiling?
So the answer is no, and I would say there are two reasons for that. One is we cloned the gene. The genetics from Konopka and Benzer was quite compelling, but it was too indirect for my taste. Jeff Hall always thought that this gene was the key, and I was less sure because I was more of a biochemist and the arguments were too indirect for me. Between cloning the gene and coming up with something mechanistic was many years. And so we were to put this another way, the sequence of the gene, we sequenced the gene in such early days, in the early mid-'80s, and there were no relatives in the database. It was a pioneer protein. It was the first of its kind. Right. And so we thought that the sequence was going to tell us what the gene did, but it didn't, because it was unique. And so we were stuck for several years. So that was one reason for not yelling Eureka. And the second reason was because even when we came up with a mechanism, we weren't sure that this was going to be true of mammals. Maybe it was just the clock in invertebrates. And of course it was because it also turned out to be true for mammals, which we did not discover. Someone else did that. We tried. We didn't succeed. That's what really gave it power. And I should say that in addition to that, the fact that it's so important for physiology also, which turned out to be subsequently the case. So, if it had only turned out to be a little bit of a timekeeper without much impact, but it turned out that so much of physiology is under circadian control, those two facts, the fact that it was true in mammals and that it governed so much of physiology, is what really gave this discovery force. And we couldn't have predicted that.
Great.
So we're just lucky. I'm a great believer in luck, in science. That's how I view things.
Yeah. I remember jumping to the ceiling with different findings that lead me to nowhere. But we're very happy.
I think I have a slide of this in other kind of presentation, but I think many good scientists are what I would call means scientists rather than ends scientists. So means and ends, I'm not quite sure what the Spanish equivalent of this is. But you can work for the ends, that is the result, or you can work because you really like the work, like an artist, like a painter.
Yeah.
And if you really like the work, then the results just come, and some they come, they go, some become significant, some disappear. Así es la vida.
Yeah, that is so true. You really forced me to learn certain really surprising things about circadian rhythm and things like that. But one thing I learned that most mammals do not share our ostensible prediction for daylight. Since what I read, only 20% of mammal species are diurnal, like us. I mean, so is there an evolutionary reason why this is so? And is there a difference between clocks of diurnals and nocturnal animals?
Right. So when I teach this to undergraduates, I think there's two major things I say about this. One is that mammals arose our origins really began in the age of dinosaurs, and mammals originally were all nocturnal to avoid being eaten by these gigantic carnivores, and so mammals began nocturnal, and then when the dinosaurs disappeared, a certain fraction of mammals then started to populate the diurnal niche, the diurnal ecological niche. So if you think of mammals as beginning nocturnal, then the fact that a relatively small percentage are diurnal makes sense. Number two, we are particularly visual. Apes, monkeys, humans. And when we got off of all four, when we stopped being small animals that are close to the ground, but suddenly, reared up so we could see ahead to anticipate what was coming and became highly visual. And of course, that's also a bit connected. Our visual system is a bit connected with diurnal behavior. Third, it turns out that in mammals, in the brain, the most important location for circadian timekeeping is a structure in the hypothalamus, which you, of course, know well, the suprachiasmatic nucleus, or the SCN. This is the major timekeeping region in our brain. And it keeps time, and it undergoes firing rate rhythms, and it fires mostly in the daytime and is quiescent at night. And that structure is identical in nocturnal and diurnal animals. So the phase, the light phase, if we can call it that, of the SCN timing is identical in nocturnal and diurnal mammals. And so the switch, the physiological switch, what is it, where are nocturnal animals different, is downstream of this major brain timekeeper. So in other words, exactly who has flipped the switch or where the switch has been flipped, I don't think we know yet. But it's not in the central brain timekeeper, it's downstream of that. So maybe in some other brain nuclei which govern behavior or something like that, some downstream connector. So a positive neurotransmitter has switched to, glutamate to GABA or something
Right.
has flipped downstream.
Mike, how do you think that this is related with part of your talk, but I think it's very important for us to learn about that, is how do you think that what we know about circadian clocks can be used to improve human health and the well-being?
Right. So I think there are two categories of answers for this. One is how should I put it? Futuristic and pharmacological. So in other words, if we could develop some medicine that reinforced or made circadian rhythms stronger, or could shift circadian rhythms instantly, so there'd be no more jet lag. All of that would be very helpful. Many blind people can't entrain because they don't have the connection with the light dark cycle, which goes through the eyes. And so if we had a way to pharmacologically with medication strengthen entrainment, or perhaps create entrainment de novo, we could both help blind people, we could make rhythms stronger in aging people who have weaker rhythms, or we could cure jet lag or help these various problems. So category one is pharmacological. Category two, like many things like this, are just good living, good, healthy habits. So for instance, I think one virtue of this time-restricted feeding is that it reinforces with food the natural circadian cycles that are happening in our body. So for example, in humans, the liver totally changes its metabolism between sleep and wake. It does completely different things. So during the day when you're eating, it's metabolizing glucose from food and actually contributing to glucose homeostasis by keeping glucose levels constant. And at nighttime, when there's no more ingestion of sugar, it's actually producing glucose. It undergoes a complete enzymatic shift to gluconeogenesis and actually synthesizes glucose, again, to keep glucose levels in the bloodstream constant. And I think by feeding and fasting, by not going to the fridge at 11 o'clock at night to have a snack before bedtime, I think we're living in harmony with these natural enzymological feeding fasting cycles of our body, and probably doing a good thing and reinforcing our circadian health in a non-pharmacological, in a physiological ways. And of course, there's another dimension to this. Sunlight in the daytime is a good thing, and light at night is not a good thing. Our ancestors experienced night as darkness and experienced daytime as full sunlight. And that is almost certainly healthy from a circadian point of view. So we have really perverted good circadian health with our indoor living, because all of us now have some kind of lighting until 10 or 11 o'clock at night, and actually are living in continuous summer, in continuous 16, 18-hour days, which is probably not such a good thing for our circadian health. So that's the sort of two categories of lengthy answers for you.
And from that point of view, have you personally changed your behavior? I mean, basis of what you know about circadian rhythm.
Well, so I don't eat. I for about the last 3 years, I don't eat breakfast. I don't eat. I fast from after dinner until lunch the next day. So I fast for about 16 hours, maybe about 16 hours. So I eat from maybe 1 o'clock to 9 o'clock or 12 o'clock until 8 o'clock during the day, and then not again until 12 o'clock. So I do that myself. I have no idea, by the way. I lost some weight, of course, but that may just be consuming fewer calories. I do that, and I try to lead a more regular circadian life. Go to bed at the same time, wake up at the same time. Don't expose. Try to avoid light at nighttime. Read and not use screens and so forth. You know, computer illumination.
Mike, this is my last question, but it's a sort of long, but it's very important for us. In the countries where daylight saving time is used, there is much discussion about eliminating 2 times during the year. And I'm going to read because I don't want to forget anything, okay? In fact, the European Union voted to eliminate daylight savings in 2019. However, there is no consensus about whether standard or summertime should be adopted permanently. All prefer summertime, as this allows them to save more light in the evening. However, this means that later sunrise, which affect children and especially adolescents, and since they are not really awake when they go to school or to the university. In your country, different proposals are being discussed in different states, depending on whether the impact on learning, standard time, or business, summertime, is taken into account. So here comes the question. What do you think is the role of science in advising decision-making about this issue?
Dios mío. So of course this falls in a bigger context, right? So you could ask, what is the role of science in decision making writ large, right? So this is only one small piece of science and decision making, right? What should the role of science be in decision making about the pandemic, right? For example, maybe that's more important these days than summertime, right? I think the science. So I should say I'm not an expert in this, but I know a little bit, and I think the science is pretty clear. That what gets complicated is the politics, like always, and what's problematic here is the science. Is it a big effect or is it a small effect? So I think the science is clear that wintertime is better than summertime. That people may not like it as much because we all like to have a drink outside at 8 o'clock at night and have our fiesta party. It's socialization, but that doesn't make it healthy. That makes it pleasant. And so it's the data are pretty clear that light in the morning is good and darkness at night is good. And I should say, by the way, that there are some very compelling epidemiological data that are very interesting and that go across a single time zone. So if you take a time zone in America that's very large, you can look at the rates of disease, cancer, for example, on the very eastern edge of the time zone and the very western edge of the time zone, where the eastern edge of the time zone has almost 1 hour more sunlight in the morning, and the western edge has 1 hour more sunlight in the evening. And the rates of disease, although the effects are small, they're consistent across all kinds of different experiments, and that the disease rates are much better when you have morning light and less evening light. And so I think from lots of points of view, you're better off with we are better off with light in the morning than light in the evening scientifically. Now the question is, like smoking a pipe, or like consuming alcohol modestly, how do we want to live, and how important the factor is. Everyone agrees that smoking 2 packages of cigarettes a day is so bad for you that it is to be avoided no matter what. But the question is a little bit of tobacco, a big deal, and so that gets complicated, and that becomes a social question. And maybe if the data get better, we will have a better sense of this. Of course, the kids could still go to school later, even if the light was earlier. That's sort of a different question. That's when do they sleep, and how do they sleep? That's sort of somewhat different issue. But I think the data are pretty clear now. Whether that, but I don't know how big an effect it is, right? And of course, then what is also clear is that the days of the switch, the very days at which things change, especially when we lose a night's sleep, as that is a demonstrable increase in traffic accidents for the next day or two. Just losing 1 hour's sleep for the population kills a whole bunch of people in the next day or two, for sure. So that's different. That's the switch. That's different if summertime or wintertime is. So that's sort of my take on the issue.
Well, thank you very much, Mike. It was a great pleasure to be with you, to see you again. And I hope that when this pandemic ends, okay, you should come to Valparaiso.
Of course I will come. I'm
with your wife, huh?
Yes, no, we were both planning to come next month, meaning we would, if the pandemic had not occurred, we would both be there for sure. So would have been there.
It was really great. It was really great. Thank you very much, Mike.
You're very welcome. Hasta la próxima.
Hasta la próxima, Mike. Good to
Ciao.
see you.
Good to see you.